Assalamualaikum warahmatullahi wabarakatuh. My name is Muhammad Faiz Hamni, and now I will explain the relationship between metabolomic and mRNA that focusing on their role in cancer biology, particularly breast cancer. So, first of all, we must know what is metabolomic. Metabolomic is the study of molecules within cells, tissue, or organism that provide insight into changes in the metabolic pathway. One of the main regulators of this pathway is miRNA and mRNA. Both is two main important key. So let's go. What is miRNA? miRNA are a small non-coding RNA molecules that act as a key regulator of various metabolic pathways. They influence cancer cell growth, proliferation, and survival. These effects are often reflected in the altered metabolomic profiles observed in different stages of the breast cancer. So, let's go for the first point. That is, miRNA as oncogenic factor and metabolomic. Some miRNA act as oncogenes, meaning that they promote cancer development. For instance, MIR3662 are examples of oncogenic miRNA. When these miRNA are upregulated, they disrupt metabolic homeostasis. So, with these disruptions, will enhance the proliferation of cancer cells by targeting genes essential for regulating a metabolic pathway, such as tumor suppressor genes. So, let's go to the second point, that is miRNA as a tumor suppressor and metabolomic. On the other hand, some miRNA can act as a tumor suppressor. For example, MIR260. They play a critical role in maintaining normal metabolic functions. This miRNA will inhibit oncogenes, leading to reduced expression in the cancer cells. By doing so, they help control the survival and proliferations of these cells. So let's go to the next slide. So, objective. The objective that we get from the study are they aim to identify specific miRNA, mRNA interactions and related metabolic pathway that change in the breast cancer. By analyzing these changes at each stages of the disease, the study seeks to better understand the key molecular events and metabolism shift, which can help to improve breast cancer diagnosis and treatment.
Assalamualaikum, my name is Faris Akmal. Now I'm going to explain the methodology. Firstly, we'll do data collection. The data regarding microRNA mRNA of breast cancer will be accessed through Cancer Genome Atlas by using TCGA BioLinks. Around 1,141 participants that have the expression data of mRNA and microRNA, each with both solid tumor and a tissue sample, will be analyzed. Then we'll do data analysis. The data analysis will be conducted using R software, which is a programming language widely used for statistical computing and graphic. Then we'll be using the ESCQ tool that help identify differentially expressed microRNA and genes between cancerous tissue at various stages and normal tissue. Next, we will be identifying microRNA mRNA interaction by using multi-MRR package to predict valid pairs of microRNA and their target mRNA. Two interactions that could happen and will be observed is downregulated mRNA that could be targeted by upregulated microRNA and upregulated mRNA that could be targeted by downregulated microRNA. Next, we will do visualization and pathway mapping. We will use Medscape plugin to visualize the relationship and interaction between genes, enzymes, metabolites, and pathway within the context of breast cancer. Medscape will integrate data from databases like KEGG to identify enriched metabolic pathway based on the expression data. Next, we will do network construction. The predicted microRNA target genes were loaded into Medscape to create a compound reaction enzyme gene network. Metabolites will be represented by nodes, while enzymatic reaction connecting these metabolites will be represented by edges. Now we will do core subnetwork. The use of Centiscape plugin to extract the core subnetwork data displaying a critical role in the breast cancer. The node centrality parameters such as eccentricity and centroid value were analyzed. Now I'm going to talk about the result from participant data analysis for each stage of breast cancer. For stage 1, we will see that downregulated microRNA such as HSA MIR1269A will affect genes like HS3S34 that cause heparan glucosamate metabolic changes that will affect glycosaminoglycan biosynthesis. For stage 2, we see that downregulated microRNA such as HSA MIR3662 will affect genes like HAS3 that cause hyaluronate metabolic changes and affect hyaluronate metabolism. For stage 3, we see the upregulated microRNA such as HSA MIR3662 will affect genes like TRHCA that cause metabolic changes such as tryptophan releasing hormone that will affect thyroid hormone metabolism. For stage 4, we see that upregulated microRNA such as HSA MIR429 will affect genes such as GDA that cause guanine metabolic changes that will affect purine degradation and metabolism. Based on the results stated, it can be concluded that the expression of microRNA either upregulated or downregulated can affect the presence of microRNA mRNA pairs and their related metabolites, which can influence the progression at each stages of breast cancer. This finding can improve the diagnosis of individuals suffer with the disease and the patient follow-up. Plus, it can also help in determining the effective drug and treatment to use.
Now, I would like to talk about the principle of biology approach in this article. The first one is cooperative analysis, which is employ a cooperative analysis of mRNA and mRNA expression between breast cancer tissue and normal tissue. This approach helps to identify differentially expressed gene and mRNAs that are associated with the various stages of breast cancer. Next one, we have system biology approach. The research utilized a system biology framework to integrate data from multiple omic layers, which is genomics, transcriptomic, proteomic, and metabolomic. This holistic view is essential for understanding complex biological interaction and pathway involved in cancer. Lastly, we have pathway enrichment analysis. The principle of pathway enrichment analysis is used to identify key metabolic pathways that are altered in breast cancer, providing insight into the biological process that may be targeted for the therapeutic intervention.
The research on breast cancer metabolomic provides several significant benefits to the medical field. So the first one is improve early detection. So the identification of specific miRNA and mRNA profile associated with different stages of breast cancer can lead to the development of more sensitive and specific diagnostic tests. So early detection is crucial in breast cancer as it significantly improves treatment outcomes and survival rate. So by utilizing biomarkers identified in this research, healthcare professionals can diagnose breast cancer at earlier stage, allowing for timely intervention. Next, personalized medicine. So the study enhanced the understanding of the molecular mechanism underlying breast cancer. So this knowledge can facilitate the development of personalized treatment plan tailored to the individual patient genetics and metabolic profile. Personalized medicine can lead to the more effective treatment with fewer side effects as therapy can design to the target specific pathway altered in a patient's cancer. Next, targeted therapies. By elucidating the interaction between miRNAs and their target mRNAs, the research can identify potential therapy target for drug development. So targeted therapies that focus on specific molecular pathway can improve treatment efficiency and reduce the risk of resistance, so ultimately leading to the better patient outcomes. And lastly, monitoring disease progression. So the research finding can aid in the monitoring of disease progression and treatment response by analyzing changes in miRNA and mRNA level over time. Clinician can assess how well a patient is responding to the treatment and make necessary adjustment. So this dynamic monitoring can lead to the more effective management of the disease. In summary, this research has the potential to transform the medical field by enhancing the early detection, personalizing treatment, and improving overall patient care in breast cancer management. So this advancement can lead to the better health outcome and a more efficient healthcare system.
Assalamualaikum and hi, my name is Muhammad bin Mushkari and today I will continue with my part, which is advantages and disadvantages. There are two advantages which are identify metabolic pathway and predict treatment response. For the first point, it will help identify metabolomic pathway that may be involved in breast cancer, which is interaction between miRNA and mRNA. The second point is predict treatment response. It also helps predict how a patient will respond to treatment. For breast cancer, the treatment response is using imaging and other tests to determine the extent of the disease and the effectiveness of the treatment, such as mammoscintigraphy. It uses a tracer to evaluate the uptake of the tumor compared to normal breast tissue. Now let's move on to the disadvantages. Okay, there are also have 2 points which are predictive value may be low. The predictive value of metabolomic signature may be too low for use in cancer screen. The second point is correlation with age. Correlation between metabolic concentration and age can be an obstacle in study of cancer risk biomarker. It's gonna be hard if the patient are too old, and it will risk the treatment for the patient. Next, limitation of research. Complexity in analysis. Based on this article, the research relies on sophisticated software tools and multi-step analysis, which may not be easily reproducible. So, for the example of the advanced computational tools such as the sequence tool, multi-MIR, Medscape, and Cityscape. For the next point, low specificity. Many diagnostic methods based on individual marker like miRNA lack specificity, reducing their standalone effectiveness. Low specificity can lead to false positives, where healthy individuals are incorrectly diagnosed as having a disease. So that's all from me. Thank you.
The database that will be used is the Genomic Data Commons GDC Data Portal, which is a unified data repository developed by the National Cancer Institute to facilitate the sharing and analysis of cancer genomic data. It plays a crucial role in supporting precision medicine by providing researchers with access to high-quality genomic and clinical data from various cancer studies, including the Cancer Genome Atlas (TCGA). The database is free to access, so it can be accessed through search engines such as Google. Just go to the search bar and type GDC Data Portal. After typing, click enter, and the link that connect to the GDC Data Portal will appear, which is GDC Data Portal homepage from National Cancer Institute. Now, click on the link and it will bring us to the GDC Data Portal website homepage. At the top of the homepage, we can see Analysis Center, Projects, Cohort Builder, and Repository. At the side, we can see various cases by major brain cells such as adrenal gland, bile duct, bladder, bone marrow, and blood. As we slide down, the website will display various other services that it can provide to us. Now, slide back to the top of the homepage and click on Analysis Center. It will bring us to a new page. Click on the plus symbol to create new unsaved cohort. Then, go to the search bar and type, and click on the first search result appear. Then a new page will appear which show list of data information related to breast disease that show information such as entity type, category, and classification. That's all. Thank you for watching.